Hello, everyone, and welcome to today's webinar, Comprehensive Meta-Analysis of Biomarker Technologies for Predictive Response of PD-1, Pd L1 Checkpoint Therapies. Presented by Dr. Cliff Hoyt, Vice President, Translational and Scientific Affairs, Akoya Biosciences, and Steve Liu of Johns Hopkins University School of Medicine. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more about our sponsor, please visit akoyabio.com. Now, before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present to you today's speakers, Dr. Cliff Hoyt, Vice President, Translational and Scientific Affairs, Akoya Biosciences, and Steve Liu, MD, PhD candidate, Johns Hopkins University School of Medicine. Gentlemen, you may now begin your presentations.
Okay, thank you, Christy, for the introduction. Hi, I'm Cliff Hoyt from Akoya. I'm the Vice President of Translational and Scientific Affairs. Before I jump into my introductory talk, just a quick overview of Akoya, in case you're not familiar with us. Akoya Biosciences is the merger of the former Akoya Biosciences, which developed and recently launched the Codex ultra-high plex fluorescence platform, with the Phenoptics platform, which we developed and supported at PerkinElmer as part of the Quantitative Pathology Solutions business unit. And so now we're one company and actively supporting and promoting both platforms, providing actually a very powerful continuum of functionality for biomarker discovery and development, which I'll talk more about now. So in the next 15 minutes, I'll cover three main topics. First, a little more detail on this discovery to translation continuum that I just mentioned, and then some more detail on the Codex platform because it's relatively new. That'll be helpful if you are not familiar with that. And then more or less an update of the Phenoptics platform because that's been around for several years now, and we've just recently introduced some exciting new functionality on that. So as many of us in the biomarker space are aware, the reason why biomarkers are so important in cancer immunotherapy is that although for many patients who receive this drug, they get lasting benefits, and this is why people are starting to play around with the word cure. The issue is that many patients don't respond, and the goal of us in the biomarker space is to develop tests that help further stratify patients into subclasses within which you get higher response rates. And a lot of the prevailing thinking these days is that the answers lie in the tumor microenvironment. How cells behave in cancer is largely a function of what's going on around them, what signals they're receiving from other cells, what's going on in the microenvironment. And the only practical way to observe what's actually going on in situ at the cellular level in the tumor microenvironment is to stain a tissue section with multiple markers and then image it. And on the right is a picture of a melanoma sample labeled with a conventional chromogenic IHC stain. You can see clearly the brown indicates there's a lot of Pd L1 protein in that sample, but beyond seeing that there's a lot of Pd L1, it's hard to tell what the actual biology is. And so by performing a multiplex stain and then imaging it, we can now instantly see an awful lot more about what's going on in the microenvironment. We can start to see lots of different cell types, what functional states are in, what checkpoint inhibitor proteins or checkpoint-related proteins are there, and so on and so forth. And once you've imaged and analyzed a sample like this, we can really start to understand that cell-to-cell biology that's driving disease progression and response. And it helps scientists start to look at things like new targets for therapeutic intervention of some sort. It's useful in trials to confirm drug mechanism of action and potentially also to stratify patients and select cohorts that are enriched for response, which is very helpful to pharmaceutical companies, especially to find effective therapies. And then potentially down the road, once these methods are validated, standardized, and satisfy regulatory requirements, help doctors make better choices and map drugs to patients in a more efficacious way. So that's the high-level view of this. At Akoya, our goal is to provide platforms that support the biomarker development continuum right from discovery right through to eventual clinical commercialization. And as you go along that path, the requirements of the platform changes quite a bit, as you can imagine. And so if we look at the sort of platform requirements as you go down that continuum, if you start on the discovery end, obviously you want to look at as much biology as possible. So ultra-high plexing is really important. A speed of analysis isn't as critical because you can enrich cohorts for response and non-response. It really is all about revealing or discovering what the driver biology is. And the biology that's going on is quite complex, so you need a lot of markers to really understand it. Also, analytically, the platform needs to be obviously good enough to reveal important biological patterns, but it doesn't have to be absolutely perfect. And that's why, as you move down that translational path, analytically, you find that you'll want to have a platform that's analytically as much as it can be so that your tests can be as sensitive and reliable as possible. Back on at the multiplexing level, as you translate from the discovery into the translational phase and trials and so on and so forth, you don't need as large a multiplexing level because you'll have discovered the biology and you'll have reduced assays to a higher throughput, more standardized assay. And then as you make your way down to the clinical end, you'll want to have reduced that assay to the bare minimum so that you can, that is sufficient, giving you sufficient information to understand that driver biology, but reduced as much as possible to keep it simple. And contrasting that on the analysis speed, you need to go from something that doesn't have to be high throughput to something that needs to be quite high throughput, and so on and so forth. So if you look at our two platforms on this continuum, Codex sits quite squarely on the discovery end. It's a very high throughput or high plexing platform, up to 50 markers today. It does have a fully automated workflow, so once the platform is set up, you can walk away and come back several hours later and have all your data. And also is quite open and flexible, so you can customize your own panels. The Phenoptics platform, although it does play in the discovery end a little bit, sits pretty squarely on the translational part of the continuum. It's a higher throughput. It includes some signal amplification to scan faster and to be more sensitive, and is really oriented around data accuracy and throughput. And so that's kind of the high-level view of where these two platforms sit on this continuum. Now I'll give you some more detail on the Codex platform. This is designed primarily for discovery, up to 50 markers today, lets you see a tremendous amount of biological detail in the microenvironment. And as I said before, it's an automated workflow. And what you're looking at up here is the what we call the Codex instrument. It's a liquid delivery system or a reagent delivery system. If you look carefully, you'll see a black insert. That's a flow cell that's designed to fit into this XYZ motion stage of several off-the-shelf high-end motorized microscopes. And so this is meant to be platform or microscope agnostic, and thus makes it quite accessible to many research labs. The basic principle here is that we reveal epitopes through oligo barcodes. So we'll have a unique oligo barcodes conjugated to antibodies, and then we have antisense oligos that hybridize to those barcodes conjugated to the antibodies. Each one of those antisense oligos has a unique color, and so we call those reporters. And the process starts with a cocktail of primary antibodies, each with its own unique oligo attached to it. So you start with a tissue section, you put the cocktail of antibodies on, and you incubate, and then those antibodies go find their respective epitopes. And then after that, there's a cyclical reveal process where 3 antibodies are detected at a time using a hybridization. And once, for example, the first 3 plus DAPI counterstain have been imaged, then those 3 reporters are removed or dehybridized, and then we reveal 3 more. And in this way, we, in a cyclical way, reveal 3 markers at a time, building up a multi-layer image up to 50 markers image. And so here's an example of I believe it's a 18-plex panel on formalin-fixed paraffin-embedded tonsil. And then after the slide has been processed through the system and all the images have been acquired, you can then view them each plane individually. And if you look carefully here and look at the markers associated with each one, you'll see that the patterns are quite appropriate biologically. And another example, this is a breast cancer metastatic lymph node. This is a fresh frozen sample, and this is a nice example to show what you can do with this data. I believe this is a 30-plex assay, and those are the antibodies in this cocktail. Once the sample has been processed and you have all the images of all the different planes, and then you've segmented all the individual cells within the image and extracted the signals in each and every cell across all markers, you can then use unsupervised clustering algorithms to identify all the various phenotypes. And you can also phenotype not only just whether they have expression, but also proximity, etc. And once you've done that, then you can overlay on top of the image what we might call a phenotype or classification map. So each one of those colors corresponds to one of the clusters in the previous slide. And you can really start to explore the biology going on in the microenvironment. And for example, this is a good example of what we call spatial biology going on in the microenvironment. This is a proximity heat map, and where you see red squares, those indicate that the 2 corresponding cell types or cell phenotypes can be found often right next to one another or very close to one another. And you can start to really explore that spatial biology in these samples. Okay, so that was a quick overview of Codex. Now I'll jump to an update on Phenoptics. Phenoptics is a platform that's comprised of 3 main components. The staining reagents, detection reagents we call OPAL, the Vectra instruments for imaging slides. These are multispectral, high-throughput slide imaging systems. And then Inform, that's the image analysis software to essentially gain knowledge from each sample. That's where tissues are segmented, cells are segmented, and the per cell data and tissue architecture information is extracted from the images. And then also, as part of the Phenoptics solution, we do have a very active contract services business in Hopkinton, Mass. the workhorse and foundation of our Phenoptics workflow, our translational workflow, is the Vector Polaris. This was a ground-up instrument designed specifically for the translational space. A lot of thought and attention went into the human interface to support a clinical-like or translational laboratory workflow with continuous load functionality and really error-free scanning. A lot of that's achieved by using carriers where up to 4 slides are loaded into each carrier, and the machine's capable of taking 20 carriers at a time, so capacity of 80 slides at a time. It's also using the latest in optics and technology, high-intensity pulsed LEDs, extremely high-performance XYZ motion system, state-of-the-art CMOS area sensors, et cetera, et cetera. And this system was developed to essentially be a digital pathology, a state-of-the-art digital pathology instrument, but also with some of the spectral technology on demand. And when I talk about multispectral, what I'm talking about is acquiring images of the sample at multiple wavelengths, not just centered on the peaks of the emissions of the floors. With that extra information, we can look at the emission fluorescence, the spectral composition of the emission fluorescence, and then do a linear decomposition to isolate all the fluorophore signals from one another and from background. This essentially gives you much more sensitive and accurate ability to quantitate the fluorescence signals. And so in contrast to how Codex works, which is an oligo-based detection approach, Opal uses tyramide signal amplification. And so the way it works is imagine a piece of tissue with 3 epitopes on it. You come in with your first primary, a first secondary conjugated to an HRP molecule. The HRP then turns the TSA fluors into radicals which covalently bind to tyrosine residues in the immediate vicinity of that first epitope. Then we come in with heat. We essentially strip or elude the primary and secondary antibodies, thus creating essentially a clean slate from a immunohistochemical point of view. And so we can come in with another primary, another secondary, a second fluor, and so on and so forth. So we sequentially layer down one color and then another so that in the end, the slide has multiple colors on it. And right now, most of our labs are doing 6-plex 7-color staining, but now many labs are transitioning to the latest 8-plex 9-color process, which I'll talk about in a second. Opal staining is now routinely done on a Bond RX from Leica. This has become a workhorse in many translational labs. It takes a manual process which used to take up to 3 days and converts it to an overnight run. So you set up the system maybe with a delayed start, and then in the morning you show up and you've got up to 30 slides stained and ready for processing. Tremendously helpful and reliable platform, and proving to be very reproducible in many of the clinical studies. There are other labs have also started using the Ventana Ultra very effectively as well. So Phenoptics has been out for several years. Last fall, we introduced some improvements or upgrades, updates. We added 2 new floors, a Opal 480, Opal 780. We also introduced a new scanning approach that greatly reduced the time it takes to do a multispectral image of a whole slide. We call it Motif. So now with the latest version of the instrument, you can scan a whole 1.5 by 1.5 centimeter area with up to 7 colors with multispectral mixing in under 10 minutes. And this is really viewed as the real translational workflow. Other things that we've done is moved away from a rectangular field-based analysis approach to a more of a hand-drawn ROI approach, more of a digital pathology approach. And then on an ongoing basis, we are sharing for free more and more of these open source R scripts with graphical user interfaces to help our research customers fully explore the spatial biology occurring in these resections. Here's an example of a 9-color assay. This is tonsil again, but labeled for Pd L1, CD20, cytokeratin, CD8, PD1, CD68, P67, and FOXP3. And if you look carefully, these are very, very appropriate staining patterns. Signals are all balanced, clean, and quite robust. And to give you a sense of what you can do with an assay like that, this is a cartoon showing tumor and its microenvironment, and then a listing of the various phenotypes that you can then identify with this set of markers. Theoretically, there's actually orders of magnitude more phenotypes that you could detect, but these are the 22 that seem to be of a lot of interest in the immuno-oncology research space. So that there are active studies and research going on that include these phenotypes. And then if you're interested in looking at the spatial information here, the black lines and the black arrows indicate many of the spatial interactions that people are now quantitating with some of the R scripts that we now offer, and also with their own R scripts. So an awful lot of information that you can get out of a 8-plex 9-color assay. And just to get a sense of how much more information you get from going from, say, a 4-plex 5-color assay to an 8-plex, if you look at the number of phenotypes that seem to be of interest based on a 4-plex, picking the most important markers from that 8, Pd L1, PD1, CD8, and cytokeratin, these seem to be the phenotypes of interest, and the black lines indicate the interactions that seem to be of interest these days. And so if you were to kind of try to capture at a practical level what the trade-off between information and plexing level, a 5-color 4-plex assay shows 8 phenotypes of interest and 7 interactions of interest. By the way, the current reagents that we sell run on NavandRx, the all-in price per slide is about $100. And if then you compare that with a 9-color 8-plex assay, generally speaking, there's 22 phenotypes of interest and 18 interactions of interest, and that costs about $800. About $200, sorry. And so one of the reasons why we think Opal reagents are becoming a standard in the IO space is because of its flexibility. In the research space, often you have clones that you prefer. You may have a lot of experience with a clone, and you want your research to have some continuity. Also, there may be clones that are accepted in the clinical market or in the clinical space you'll want to use. So you can, because you can pick your own clones with Opal, you can then pick whatever clones you would like. Another key feature of Opal approach is that you can amplify to suit your research goals. And that's really important in IO, especially with checkpoints, because often you really want to be able to see even the very low expressing cells. Another aspect of the flexibility to amplify according to your research is expression levels vary from tissue type to tissue type, so you may want to optimize differently for different tissue type. Another aspect of Opal that seems to be quite appreciated is that once you've done a couple of these panels, it's easy to mix and match. You can swap in and swap out different clones. And so onto the newer image analysis workflow supported by this whole slide multispectral capability. Here's an example of a lung cancer sample stained with the new Opal dyes and then scanned with this new motif functionality. The upper row show heat maps of densities of the various cells revealed by these markers. The lower row shows some of the more complex assessments. The 3 on the left show density of double positives, and then the 3 on the right show the density of instances where one cell of interest is within 30 microns of another cell of interest. So this is where you really start to look at the spatial biology. And if you think there's a particular immune suppression mechanism going on related to the interaction between two cells, this is apparently the most effective way to really understand if that's happening. Another example, and this, by the way, is also supported by one of the R scripts that we now share. You can draw or annotate a tumor, and then the software will then draw essentially shoreline plots around that boundary, and then read out the density of the various cell types as a function of distance to that boundary. And you can really start to see some interesting biology and see things such as immune suppression or immune infiltration or immune desert in a quantitative way this way. And finally, my last slide, we thought it might be helpful to put together just kind of a laundry list of the things that you might want to consider when you look at the various approaches to doing multiplexed immunofluorescence. Obviously, multiplexing is very important, depends on where you are on that translational continuum. Sensitivity and dynamic range, pretty important in the discovery end because you obviously need to be able to see things, but that becomes extremely important as you move into the trials and studies, and especially as you move towards the more translational end. Reproducibility, same thing, very important in the translation study trial space. If you discover something important, you want to know that it's really robust. Same thing with validation, very important in the translational part of the continuum. And in particular, as you move down that path, you'll probably want to show that you're at least as sensitive to the gold standard, which is optimized chromogenic DAB IHC. Flexibility, more important on the discovery end, and then some more practical issues such as staining and imaging rate. How many slides can you stain and image a day? Image analysis rate is turning out to be really important, especially as you scan whole slides. It takes a long time to analyze a whole image, a whole slide image actually. Very practical things such as cost per slide, cost of the platform, ease of use. And then the last bullet is really important too. If you're going to invest a lot of time in discovering and developing a biomarker, you'd like to know that there's a potential future where that test or biomarker could potentially be translated into the clinic for the benefit of patients. That was helpful, and now I'd like to hand this off to Steve. Thank you very much.
Thanks, Cliff. Hi, I'm Steve. Thank you for the opportunity to present today. So today I will be presenting a meta-analysis comparing the relative diagnostic performances of several commonly used biomarker modalities for predicting response to anti-Pd L1 monotherapy. I have no disclosures. So with the success of anti-PD-1 therapies, there has been substantial effort ongoing to identify predictors of response to anti-Pd L1 immunotherapy. Numerous different biomarker assay modalities have been proposed, and several have actually been shown to be associated with response to anti-Pd L1 when tested on pre-treatment tumor tissue samples. However, the relative sensitivity and specificity of these different approaches have yet to be systematically established and reviewed. So the studies that I'll be talking about today include Pd L1 IHC, which, as many of you know, is the most commonly used and well-established of all the modalities for immunotherapies. I will be also looking at studies that look at tumor mutation burden, which they use as a measure of tumor antigenicity. I'll also be talking about studies that use RNA for gene expression profiling panels. And I will also be looking at studies that have used multiplex immunofluorescence, IF, or IHC, to quantify multiple proteins simultaneously within the tumor microenvironment. I will also be going into some instances where investigators have combined multiple of these approaches together, which I will call multimodality. And so throughout this meta-analysis and this presentation today, the key question will be, what are the relative efficacies of these different biomarker approaches for predicting response to anti-PD-1 monotherapy? So for today's talk, I'll start by describing how the studies and trials that are included in the meta-analysis were initially selected and how the individual data points and values were extracted. Then I'm going to go into the detail of the meta-analysis itself and how we determined the diagnostic performances of the different modalities. And this will include how we generated the curves to calculate area underneath the curves and other statistical measures. I'll then discuss some of the additional analyses that we did to address certain issues such as how we treated stable disease, as well as OS and progression-free survival. And then lastly, I'll talk a little bit about how various multiplex approaches that were included in this study and how they were, and use Merkel cell carcinoma as an example. So first, I'm going to start by discussing how the studies were initially filtered and excluded. So here's a schematic showing briefly the selection of all the different biomarker studies that were included. So publications were initially identified through a PubMed search, and then additional studies were identified through a search of meeting abstracts such as ESMO, SITC, AACR, or ASCO. And also, just due to the large number of studies that have used Pd L1 IHC, we actually only included studies that resulted in FDA approval that were indicated on the NIH's Daily Med website. This led us to about 243 records ultimately being screened. And then we ultimately excluded 198 records. And so this included for reasons such as that the studies were either too small, so we only included studies that had at least 15 samples or greater. Either there was no corresponding response or PFS or OS data. We actually only included studies that looked at solid tumors and included none of the liquid tumors. And then we excluded any blood-based profiling studies. And most importantly, we made sure that because there are so many studies that can overlap even within the same cohorts, we made sure that each patient sample was only represented once in the meta-analysis so that there were no overlapping patient samples. So this led us ultimately to 45 unique publications with 56 individual analyses that we could do. Over 10 tumor types were represented ultimately, and this led to 8,135 samples that were uniquely represented within the meta-analysis. So for the studies that were included, we tabulated the median age, sex, tumor stage, and tumor types between the various assay modalities. Here, the Pd L1 IHC studies are represented in blue, TMB in red, gene expression profiling in yellow, multiplex IHC-IF in green, and multimodality in purple. So when we tabulated all these together, we saw that in general, the clinical covariates did not seem to vary significantly between the assay modalities. The majority of our studies, about 96%, included tumors that were advanced stage 3 and 4. And as expected, because Pd L1 IHC is the most commonly used of all the modalities, and it is actually well represented across many of the tumor types. However, for the other newer kind of 4 modalities, such as TMB, gene expression, multiplex, and multimodality, they are relatively evenly distributed between urothelial, non-small cell lung carcinoma, and melanoma, which is not necessarily surprising given that these are the major tumor types that have the kind of the largest trials and studies ongoing. So with each individual study, we then needed to extract the individual patient outcomes and correlated it with each patient's test results. This ultimately allowed us to generate a 2x2 diagnostic table that included whether or not the patient was a responder or non-responder versus whether that assay test was positive or negative. This then allowed us to calculate a sensitivity and specificity for each individual study that could then be plotted on a 1 minus specificity plot that we see on the right. So before I go deeper into the meta-analysis itself, I wanted to talk about several considerations that occurred when we were extracting the data from the studies. So there were some things that I wanted to note that I will also go into further detail a little bit in the talk as well. So one of the things that we encountered was that even though Pd L1 IHC is one of the most commonly used assays, there is still a large variation in how the test is assessed and performed, even between institutions. For example, as many of you already know, there are different antibodies clones that are being used. The percent positivity could vary between even tumor types and even just between studies. For Pd L1 IHC specifically, part of how we tried to address this was by using FDA-approved cutoffs whenever possible. And another example of test variability would be for things like gene expression profiling, where the number of RNA analytes could vary very significantly, whether it be a very small panel of genes in one study versus another study which may use a very large panel of many genes and many signatures. Secondly, there were actually a few studies, about 10%, that only had corresponding disease progression or survival data. And when this was the case, we actually selected the time point that provided the highest sensitivity and specificity value. And how we did this was by using the Uden statistics. The Uden statistics is a measure of informedness, which allows you to select the optimal point cutoff that maximizes sensitivity and specificity. And so therefore, this allowed each study to have the highest possible performance value and each study to perform maximally within the meta-analysis itself. So to compare the relative performances of the different biomarker approaches, we plotted each study's value on a sensitivity or true positive rate on the which is shown on the y-axis versus a 1 minus specificity or a false positive rate, which is represented on the x-axis on this graph. So just for reference, an ideal test is going to be on the upper left-hand corner, and this ideal test would have 100% sensitivity and 100% specificity, meaning a 0% false positive rate or 1 minus specificity. Here, what I'm showing is all the sensitivity and specificity values for the Pd L1 IHC studies, and these again are represented in blue. The size of the dots represent the size of the cohort within each study for that particular assay. So the larger dots represent larger studies. And just to reiterate, again, each study had the maximal opportunity to perform with the meta-analysis. So here now I'm showing the remaining specificity and specificity values for the studies for the other modalities. Again, TMB is represented in red, gene expression is represented in yellow, and multiplex IHC-IF is represented in green. So now I'll go further into the actual meta-analysis itself and how we compare the diagnostic performances of each modality. So to perform the meta-analysis, we generated summary receiver operating characteristic curves, or summary ROC curves. This allowed us to determine a relative area underneath the curve, or an AUC. The AUC then served as a global metric of each approach's ability to discriminate between responders and non-responders to therapy. On the left, I'm once again just showing the points for Pd L1 IHC represented in blue. Once a summary ROC curve was generated, the calculated AUC for Pd L1 IHC was determined to be 0.65. Using this random effects model that we used to generate the curves, we could then also calculate a 95% confidence interval for each AUC, which is represented in the dotted blue line for Pd L1 IHC. I'll go into a little bit later, but we were also able to do this for individual tumor types, for non-small cell lung carcinoma, melanoma, and urothelial carcinoma. So here I'm showing the summary ROC curves that were generated actually using two different approaches. On the left is one approach that we're going to call weighted, where the size of each study is taken into account. So the larger studies that have more patient samples would hold more weight than a smaller study. On the right, we used another approach, which I'm going to call unweighted, where each study was treated the same regardless of size. And so on the left, with the weighted approach, as I had mentioned earlier, Pd L1 IHC had an AUC of 0.650. TMB, represented in red, had a slightly higher AUC of 0.688, while gene expression profiling had a similar AUC as Pd L1 IHC, with an AUC of 0.650. Interestingly, multiplex IHC-IF had the highest AUC with a value of 0.790. Interestingly, on the right, we see that the trends seem to hold the same even when the studies are treated equally, with TMB in red, gene expression in yellow, and Pd L1 IHC in blue, all seemingly clustered together, with multiplex having a much higher AUC represented in green of 0.878. So specifically, the AUC of multiplex was significantly higher than the AUC of Pd L1 IHC, gene expression, and TMB. And actually, the AUCs of Pd L1 IHC, TMB, and gene expression profiling did not statistically differ from each other. And just as a reference, it has been generally accepted that for diagnostics used for patient selection, the test should have an AUC in the range of about 0.8 or above to be clinically useful. So for the next concept, I wanted to discuss the multimodality studies, which are going to be represented in purple. These are studies where investigators have combined multiple biomarkers to predict response to see if there's any added potential benefit. And so because these are very new, there are definitely fewer studies. And on the left, I'm showing the multimodality studies, again in purple, overlaid on top of the multiplex studies, which are represented in green. The multimodality approach, the summary ROC curve, had an AUC of 0.736, which began to approach that of the multiplex summary ROC curve. And so one thing to keep in mind when thinking about these multimodality approaches, though, is the ease at which these approaches can be implemented in clinic. Pd L1 IHC may have the lowest AUC out of all kind of the modalities, but it is relatively easy to implement. On the other hand, incorporating multiple tests together will be costly, and in order for these approaches to be implemented in the clinical setting, they must be significantly better than the other methods in order for it to be practical. So now I've also overlaid the original Pd L1 IHC, again in blue, TMB in red, and gene expression curves in yellow. And interestingly, we see that compared to the AUC of Pd L1 IHC alone, TMB, or gene expression alone, the purple curve for the multimodality had a higher AUC than any of the three alone. So this supports the concept that an inflamed tumor microenvironment, whether that's represented by Pd L1 IHC or by gene expression profiling, is distinct from potential tumor antigenicity, which is represented by TMB. And so when combining these together, these tests together, there could be additional predictive or potentially even prognostic value that should be explored further. So now I wanted to look at the performance of the different modalities in a slightly different way. Even though different approaches might have similar AUCs, they actually might have different predictive values. And so what I've plotted here are the negative and positive predictive values for each of the studies in the meta-analysis. Again, the size of the dots represents the size of the studies, similar to before. So for reference, an ideal test is going to be 100% positive predictive value and 100% negative predictive value. So it's going to be on the top right-hand corner, which I've indicated with a star. If we were to divide this graph into 4 quadrants, we can already begin to see some patterns emerging. So most of the modalities most of the modalities seem to have a high negative predictive value, and so are clustered towards the top half of the graph. So this means that they're good at excluding patients from treatments with a negative result. However, the multiplex IHC-IF studies, the green dots, also seem to have a generally higher positive predictive value and seem to be more up in the upper right-hand quadrant. And so this means that they are potentially better at identifying patients that should be on treatment and would benefit based on a positive test result. And so actually these trends seem to hold even when looking at individual tumor types individually. So another way I wanted to talk about each biomarker approach was using likelihood ratios. The advantage of likelihood ratios is that it does not depend on prevalence, whereas it does depend on prevalence for predictive values. So the likelihood ratio value combines information about both sensitivity and specificity. And in the predictive biomarker setting, it gives an indication of the odds that a patient will respond or not based on their test results. So on the left, I have a plot showing the negative likelihood ratio on the y-axis and the positive likelihood ratio on the x-axis. For reference, an ideal test is on the bottom right with a very high positive likelihood ratio and a negative likelihood ratio near 0. Once again, if we divide the plot into quadrants, we see trends begin to emerge. So the multiplex IHC-IF studies, represented in green, seem to generally have better negative likelihood ratios and higher positive likelihood ratios. And when we actually pull these together, which we see on the right, we see that the multiplex and the multimodalities approaches had a significantly higher positive likelihood ratio than Pd L1 IHC alone, and that the multiplex IHC-IF studies also had a better negative likelihood ratios compared to the other studies. So now I wanted to go to some of the additional analyses that we did for the meta-analysis and talk about some of the challenges that we encountered during the analysis. So for the analysis, stable disease was initially treated as a non-responder. However, what would happen if stable disease was treated as a responder? So a portion of our studies actually had individual patient data that allowed us to manually regroup stable disease into different categories. And so out of these 18 studies, on the left, I have all these studies with their plotted sensitivity and specificity values and the corresponding summary ROC curves that were generated. If we were then reanalyze the stable disease and put them into the responder group, we get the summary ROC curves on the right. And I'm showing how the dots slightly shifted compared to how we put the stable disease. However, we see that actually the trends seem to be very consistent. And so this suggests that stable disease, grouping stable disease has very little impact on the summary ROC curves itself, and that the diagnostic performances of these modalities appears to be quite stable. So as I had mentioned before, there were about 9 out of 56 analyses, so about 16% studies that had responses extracted from OS and PFS data. And so these points are highlighted with arrows. And so what happens if we were to remove these studies from the meta-analysis? So on the left, it's what we have seen already before, where all of the studies are included. However, on the right is where the studies with an OS or PFS data were excluded. And so these are the new summary ROC curves that were generated. And similar to what we were seeing with stable disease analysis, we show very similar trends between all the different analyses, and the AUCs seem to be very comparable. So the results were also consistent when it was either weighted or unweighted. So we see that the results seem to be very stable, regardless of how we reanalyze the data. So another one of the challenges that we encountered when we were doing this meta-analysis was with the TMB studies. So similar to Pd L1 IHC, there are different TMB cutoffs between different tumor types in different studies. So one of our collaborators, who happened to be an expert in the TMB area, suggested that we reevaluate the TMB data using a tertiles approach. We were able to get raw data from 6 out of the 10 TMB studies and created a more uniform thresholding strategy where we use the upper tertile or greater than 67% as a test positive or high TMB versus the lower tertiles. And using this method, it allowed us to potentially account for any test variability within each study. And so what we see is a new summary ROC curve that was calculated, shown in pink, and the original one, again, shown in red. We see that the two curves seem to be quite similar and that the AUCs seem to be quite stable, regardless of how we thresholded the TMB cutoffs. So I also wanted to note that for this meta-analysis, we actually did not include any MSI studies into the TMB category. So the reason for this was that we believe that the biology is very different. In MSI, there are a significant number of indels, where compared to other cancers, investigators were typically measuring non-synonymous mutations. It's interesting to note, so the MSI studies that we included, which is represented very close to the upper left-hand corner, is that the MSI studies actually performed very similar to that of the multiplex IHC-IF. So what happens when we look more closely at individual tumor types? Do the biomarker patterns still hold true? So we looked at non-small cell lung carcinoma, melanoma, and urothelial carcinoma, as those were the 3 main tumor types that had enough studies across most of the modalities and to allow us to do a meta-analysis. So when we looked at non-small cell lung carcinoma, the trends seemed identical to that of the pan-tumor analysis that we did before, where the multiplex, again represented in green, seemed to have had a much higher AUC than either the Pd L1 IHC, TMB, or gene expression studies. In melanoma, gene expression studies, which are represented in yellow, seem to actually have a comparable AUC to multiplex, which is very interesting. However, actually, only the multiplex IHCIF AUC was shown to be statistically significant compared to the Pd L1 IHC alone. So this was likely due to the fact that the gene expression studies had more variability in the sensitivity and specificities. So the confidence intervals were much larger for gene expression. For urothelial carcinoma, there were not enough studies for multiplex to generate an AUC. But interestingly enough, the gene expression approaches actually seem to perform the worst compared to Pd L1 alone or TMB. However, the most important thing was that the AUC of multiplex and multiplex IHCIF was significantly higher than that of Pd L1 IHC when compared with all tumor types, which we saw before, and for non-small cell lung carcinoma or melanoma alone. So actually recently there was another meta-analysis published in JAMA Network Open journal where they looked at lung cancer and the response to immunotherapy versus conventional therapy. So in this study, the authors actually included a sub-analysis where they combined 3 non-small cell lung carcinoma cohorts that had individual patient data. And then they evaluated how TMB, PD-L1 IHC, or a combination of them performed at predicting patient responses. So actually very similar to our results, they saw that TMB itself had a higher AUC than PD-L1 IHC alone, and that PD-L1 IHC alone seemed to perform the worst. And by combining TMB and PD-L1 IHC together, so like multimodality, which we saw before, that actually had a higher AUC. And so they saw this both actually with objective response, which is what we have been looking at, which we were primarily looking at, but they also had progression-free survival data, and so they saw the similar patterns emerge. So lastly, I wanted to end with a discussion about the multiplex approaches that were included within a meta-analysis and use Merkel cell carcinoma as an example. So I hope to have now shown that the multiplex IHCIF studies seem to generally demonstrate a higher AUC than the other modalities that is consistent across many tumor types. And so the reason that there is this better performance is likely due to the fact that by staining multiple markers on fewer slides, you're able to get co-expression information, spatial relationships, and also proximity and distance information about the tumor microenvironment. And because we know that the immune response is very important for whether or not a patient is going to respond, it's not unreasonable to expect that higher resolution of the tumor microenvironment would likely lead to more insight about a patient's response to therapy. So on the left, I'm showing the multiplex IHCIF studies that were included in our meta-analysis, and I've annotated each of the points with the biomarker combinations that were used for each study. So as you can see that it is actually quite a heterogeneous group of markers, but we see that the majority of the studies actually do include CD8 and or PD-L1 as a marker. And then some studies also looked at T-cell functionality, such as with granzymes, and then other studies looked at distance and spatial relationships. So what will be very interesting to see is whether, as there are more multiplex studies being published, if there are going to be any combinations that are going to be superior to others for predicting response. And we can already see on the left-hand side that there are some studies that some biomarker combinations that seem to have very high specificities, which are going to be represented on the y-axis, and then also some combinations that have very high sensitivity values. And so those are the ones that are going to be on the top of the graph on the x-axis. And so it will be interesting actually also to see if combining some of these markers which have very high sensitivities and very high specificities, if that will actually yield even better combinations that will be in the upper left-hand corner. So here I wanted to go into an example of two studies that were included within our meta-analysis that looked at Merkel cell carcinoma. One of the studies was a Pd L1 IHC study, and the other was a multiplex IHC-IF study. So it has been reported for Merkel cell, as well as other tumors, that there are examples of constitutive Pd L1 expression in the tumor microenvironment, and that this Pd L1 expression is not associated with immune infiltrates. So on the right, we have two patients. Both are going to be positive for Pd L1 IHC. However, only one of them has corresponding PD-1 expression, suggesting that only that patient has an active anti-tumor immune response. And so only the top patient is actually going to be the one that will likely respond to therapy. However, because both of them would test positive by just Pd L1 IHC alone, it would likely that the patient currently on the bottom might be given treatment, but would likely not respond. And so this is an example of why there is a potential proportion of patients that have PD-L1 positive tumors, but do not respond well to anti-PD-1 therapy, as it is more the adaptive as opposed to constitutive PD-L1 expression that indicates an endogenous anti-tumor immunity. So I wanted to highlight how one group, a study that was done by Janice Taub's lab that was published in JITC last year, tried to determine whether or not a patient has an adaptive response to the tumor. They did this by measuring the close proximity of PD-L1 positive cells in the tumor microenvironment to tumor infiltrating lymphocytes. And so they saw that the density of PD-1 positive lymphocytes that are adjacent to PD-L1 positive tumor cells correlates with clinical response to anti-PD-1 therapy. So the density of PD-1 positive cells adjacent to PD-L1 positive cells was actually significantly higher in responders versus non-responders, as one may expect. Interestingly, when this proximity parameter was normalized for individual PD-1 or PD-L1 cell densities, the association was preserved. So this actually suggests then that the proximity aspect could add some additional value to the predictive capacity of PD-1 and PD-L1 densities alone. So I hope to have shown now that this meta-analysis, we saw that PD-L1 IHC, although it's one of the most well-established biomarkers for response, actually has a poor likelihood ratio and the lowest AUC compared to the newer modalities. And also, I hope to have shown that a multimodality approach may provide increased benefits compared to a single modality approach. And I hope to also have convinced you all that the newer multiplex IHC-IF strategies seem to have generally yielded better positive and negative likelihood ratios with a high AUC that was approaching 0.8 or higher. And so lastly, I wanted to end on that this whole biomarker field is constantly a work in progress. So already with T and B, we see that not all mutations are equal, that now investigators are focusing on neoantigens, that some mutations potentially are better than others, and that some mutations in certain genes, such as STK11 or in the Wnt pathway, might play an important role in whether or not a patient is going to respond or not. Furthermore, with gene expression profiling, we see that there's the expansion of gene signature panels, where initially it was a very small panel looking at specific interferon gamma genes, but now it's expanded into very large inflamed gene signature sets. And even with multiplex IHC, although the multiplex studies we're looking at included only a few markers, now the panels have expanded from 6 color panels to even larger color panels. And so that will potentially provide even more information about the tumor microenvironment and allow one to see even more spatial and different cell types within the tumor. So lastly, I wanted to thank you, Akoya, for the opportunity to speak today. And I wanted to just thank several people who have been instrumental for the study, including Luda from Johns Hopkins and Daphne and Julie, as well as Janice Tao for their support on this meta-analysis. And then I'd also like to thank some of our collaborators, Kurt Schauper and David Rim from Yale, as well as Matthew Hellman from Sloan Kettering, and then also Douglas Johnson from Vanderbilt, as well as Jeffrey Sossman from Northwestern. And of course, I'd like to thank Cliff for his help in supporting this study. And so now we can open it up for questions in our live Q&A.
Thank you, gentlemen, for your informative presentations. Now let's begin our live Q&A. If you have a question, just click on the Ask a Question box located on the far left of your screen and click Send. I will now pass this over to Chris Streck, Vice President of Marketing, Akoya Biosciences, to facilitate our Q&A.
Thank you, Christy, and I'd like to thank both of our presenters, Cliff and Steve, for a very exciting and informative presentation. As a quick note, since we're coming up towards the top of the hour, we're going to focus the majority of the questions for Steve and related questions around the study. Any questions that come in that we might not get to, we will be happy to follow up via email. I'd also like to note we've been joined by one of our application scientists, Bethany Ramanik, who can also answer technological questions. So the first question is for Steve. What criteria was used to select the studies for your meta-analysis?
Yeah, hi, Chris. So the initial studies were just identified through PubMed, kind of with the usual keywords for all the PD-1 and Pd L1 monotherapies. However, kind of one of the issues that we encountered when doing this this initial search was that, as I had mentioned kind of earlier as well, was that there was just a huge amount of studies that have done Pd L1 IHC studies. And so one way that we thought was a reasonable approach to kind of focus through all of the studies was to use only those studies that were indicated on the NIH's Daily Med website that resulted in FDA approval. And so that was kind of how we helped really narrow down the large body of work, especially in Pd L1 IHC.
Great, thank you. How did clinical covariance influence the data, or did clinical covariance influence the data in any way?
So the clinical covariates that we looked at were the median age, the percentage, the males, the sex, and tumor stage and tumor type. And so in regards to, when we looked at those four things, we did not see any variation between, no statistically significant variation between each of the five modalities. And they seemed pretty uniform across the board. I think it was often because a lot of these studies that were either with the newer modalities were coming out of those larger Pd L1 IHC cohorts, and so I think in general the overall variation was relatively the same.
The next question, again for you, Steve, is how is the positive Pd L1 identified in a multiplexed IF study, and does that take into account other markers such as CD8 expression or distance?
So in those that were in multiplex, they did some studies, many of the studies took into account Pd L1, but it was always Pd L1 in relation to something, and that would, as it was annotated on the earlier slide, could vary between study to study, whether it be the distance or whether it be to a tumor marker or whether it be to tumor infiltrating lymphocytes. And so I think I was trying to highlight that usually Pd L1 or CD8 is included in these multiplex studies, but it's always in relation to something else. And so because it's a newer technology for sure, the hope is that as more and more studies come out, that will kind of narrow down some of the more, hopefully more successful combinations.
Great, thank you. So based on these results or other studies that you may be part of, do you think that these same results will hold true for other cancers such as breast or colorectal?
So I think So I obviously will have to see what the results are going to be. I think it's pretty clear how Pd L1 IHC is performing across many of the different tumor types. I think it's, as I mentioned earlier as well, I don't think it is unreasonable to expect that some of these methods, such as at least multiplex and potentially even gene expression, that where you're getting multiple inputs and you're kind of getting this higher resolution of the tumor microenvironment instead of just kind of one single readout for the tumor response, is you're likely going to get hopefully better predictors of response. So I would expect hopefully they will at least perform equal to Pd L1 IHC, but hopefully I expect that they would perform probably better as well in the other tumor types.
That's great. And building off of that, based on this study, what considerations should we take into account for moving forward with other clinical research, in your opinion?
So if I am understanding this question correctly, it's saying like how should we kind of take how should we look at the biomarker field? So I think that this study has hopefully systematically organized and reviewed all these different methods in one place and make it in a digestible manner. I think what's clear is that Pd L1 IHC, although it's very easy to use and has worked for some amount of time, that there's clearly still a lot of work to be done. Whether it's going to be multiplex, that's the final answer, or something else potentially with a lot of these newer methods that are coming like CyTOF, will remain to be seen. But I think it shows that clearly there is a lot more that needs to be understood and can still be identified with the tumor microenvironment that will be definitely better markers for patients.
Great, thank you. All right, well, I want to again thank our speakers for the great presentation, and Steve, it was very informative, and appreciate you standing by to answer questions. Given we're about 10 minutes over, we'll close the questions for today, but we have a large number of questions both about the study as well as the Akoya technology. Again, I'll remind our listeners we'll respond via an email with detailed answers to some of the questions we didn't get to in the verbal portion of our call today. So with that, I'd like to thank everyone who's joined. We've shown the contact information on this last slide. Please visit us at akoyabio.com or send us a direct email at info@akoyabio.com. And as a last reminder, if you'd like to listen to this presentation again, very shortly after the event, we'll have it posted up in an archive form where you can review it at your leisure. So thank you again, and everyone have a great day.